Snap connection. We will have a new set of questions, image-based questions, and a connection between them for the episode 3. And also we'll quickly discuss the answers of prior episode since many of you had asked the same. Let's go ahead with episode 3. The rules are the same. You need to identify all the images as a whole. There are 4 images today again, and you need to identify the connection between them. As already mentioned, you need to answer the contest within 24 hours in our YouTube comment section or in our Instagram page. And we have a bigger prize this time. Let's see who gets this. This is our image number 1 for episode 3. There's a small clue at the top, autosomal dominant condition with the image you need to identify. This is image 2 for today. A CT image, sagittal section showing something. You need to identify the same. This is image 3 for the episode number 3. There is something abnormal that you need to identify in these images. Axial flare image and coronal flare image of MRI brain showing something. This is your image number 4. It's a bit difficult, but if you have got prior 3 correct, this one will be a cakewalk. They are showing something, expression of something which is required for this region of the pons and medulla to function, expression of a gene, and if that gene is not functional, it will lead to something which will cause improper functioning of this portion of pons and medulla. You need to identify the disorder, not the gene as such. You need to identify the disorder. Once you get that right, you'll be able to connect all 4 and answer it very quickly. You can post your comments in the comment section. We'll move on to the answers of episode number 2. The first image was of Duchenne muscular dystrophy. We initially thought we would go ahead with Gover's sign, but it would have been too easy for everyone to answer. So we showed MRI of bilateral lower limbs, and you can see muscles atrophied, fatty atrophy of the muscles of both sides with relative sparing of few muscles, biceps semimembranosus. So that was the clue. This was the image number 2 of the prior episode number 2. As you can see, tense blisters, few of them of different ages, leaving scar have been shown in the image, and the main clue was hidden in the immunofluorescence study here. You can see this was called for immunofluorescence study. You can see the blister here, and the important thing to note is that from your histopathology you would have seen that there are 3 types of epidermolysis bullosa: simplex, junctional, and dystrophica. Simplex, there is affected keratin 5 and 14. Then there is junctional, where laminin is affected, and in dystrophica, where type 7 collagen is affected, and you can see where the type 4 collagen lies. Type 4 collagen lies in lamina densa portion. So what happens if there is a blister In dystrophica epidermolysis bullosa, what happens is the blister will be deep to this lamina densa layer, and hence type 4 collagen, which makes most part of the lamina densa, will be stained here at the roof of the blister, while the other two, erythema bullosa simplex and junctional epidermolysis bullosa, will have staining of the floor of the blister. So this was an important clue. Other than that, if there is a patient with this tense blisters, you need to remember this important approach. Next, in the image number 3, next image of episode 3, we had given this particular diagram. It seemingly looks normal, but what you need to appreciate is there is dilated membranous labyrinth. This is how the normal labyrinth should be. However, it is dilated here and it is seen in one condition, Meniere's disease. So you need to remember that, which leads to a lot of symptoms including tinnitus and vertigo. The last image was of this particular image where it showed gram stain image of organism, gram positive organism, which had safety pin appearance or bipolar staining, though it was difficult to identify because there are quite a number of organisms showing safety pin appearance. You need to also, safety pin appearance is better appreciated in peripheral smear. So you need to remember that once you link all the 4 images, you'll get the answer, this particular image easily. So the link was aminoglycosides. Most of you would know the infectious uses of aminoglycosides, while it is drug of choice for one of disease, that is for few diseases, one of which is your Yersinia pestis, and the only organism which has aminoglycosides as drug of choice and with a safety pin appearance. You would get that part easily if you had got the other 3, but most of you would not have been aware of this particular thing, that aminoglycosides were tried in the initial part of 2010s for its application of codon read through in conditions having stop the nonsense mutations, which lead to stop codon being inserted in a protein in a gene. So what happens is that protein would be cut short with few amino acids not read through. So instead of that, what aminoglycosides do is based. So as you can see in this, a stop mutation leading to synthesis of replacement of a stop codon at a particular site leads to truncation of this protein with fewer amino acids being synthesized. However, what aminoglycosides do is, instead of reading this stop codon as a stop codon, it allows the complete read through of a protein, hence allowing synthesis of a normal protein. This was initially tried in Duchenne muscular dystrophy. However, it was withdrawn. But however, latest reports suggest that aminoglycosides have been successfully used in treating dystrophica epidermolysis bullosa, and it's in currently phase 3 trials and it will soon roll out. Another important thing you need to remember is the side effect of gentamicin, which is its vestibulocochleotoxic nature, has been employed in Meniere's disease in retractable Meniere's causes. So connecting all these images, you could get through an answer. So today's questions are bit trickier, but we will definitely help you out with a few clues. If you are unable to get it within 24 hours, try to get it within 24 hours, and hopefully you have enjoyed this session. Thank you.